So today's video is going to focus solely on the plasma membrane. So we've talked about a variety of other organelles, and so today our focus is going to be on the membrane only and how that structure of the cell membrane is what allows it to be semi-permeable and to only allow certain things to pass back and forth. So let's first take a look at what the cell membrane is made up of. So we're going to start with our phospholipids, because our phospholipids are going to they're going to be what makes up the bulk of the membrane. So all of these blue molecules here, all of these are phospholipids. And the phospholipid is what we call an amphipathic molecule. So it's pathic. So these phospholipids being amphipathic means that they have both a hydrophobic and a hydrophilic end. And so the hydrophilic end is going to be the phospholipid head there. So that would be the hydrophilic portion of the phospholipid. Okay? And that hydrophilic portion is going to be attracted to water. It contains a phosphate group, and because it contains a phosphate group, that gives it a charge. And so that is what makes the hydrophilic end like water, because remember, water has a charge. It's a polar molecule. The tails, the little squiggly lines here, so the tails here, these are the hydrophobic portions of the phospholipid. They are hydrophobic, meaning they don't like water, because these are made of fats. Okay? This is the lipid part of the phospholipid. So the hydrophobic end is made of fatty acid chains, and because it's made of fats, therefore it's nonpolar, they arrange themselves towards the middle here so that they are away from the aqueous layer. And so because of it being amphipathic, so amphipathic means that it has both its hydrophilic and hydrophobic parts. Because of the cell of the phospholipid being amphipathic, it's going to form this bilayer. There's always going to be these two layers. The nonpolar fatty acids arrange themselves towards the center, where the hydrophilic phosphate groups, or the phosphate heads, can point out towards the aqueous environment. So those are your phospholipids, and that's what's going to make up the bulk of your cell membrane. So now we're going to talk about your different kinds of proteins that could be in the cell membrane. So the proteins that are within the cell membrane, they can be hydrophilic or they can be hydrophobic. Okay? And if they're hydrophilic, they're going to have charged polar side groups. So if the protein is hydrophilic, it's going to have a charge. If it's hydrophobic, it's going to have nonpolar side groups. Remember, all of your amino acids have that NCC pattern, right, where they have the R, which is what makes the amino acid different for the various amino acids, right? And proteins are a bunch of amino acids linked together. So if it's a hydrophilic protein, these R groups are going to be polar. If it's a hydrophobic protein, those R groups will be nonpolar for those various amino acids. And we'll talk more about transfer proteins and channel proteins as we move into how the membrane actually allows things to move back and forth. Right now we're focused solely on the parts. So these would be examples of proteins. Here's another example of proteins down here. So now your next molecule that can be in your membrane would be your steroid molecule, or cholesterol. The steroid molecule is these little yellow ones that are embedded in here. Right, there's our steroid molecules there. And those steroid molecules are going to be found in animal cell membranes. So the steroid molecules are in animal cell membranes only. And these cholesterol or steroid molecules in there, they're going to affect the fluidity of the membrane. And what it means by that is that they are going to affect how rigid that membrane is. And so at very high temperatures, the cholesterol makes the membrane less fluid. These phospholipids, they can move back and forth. They don't usually flip from one side of the membrane to the other. They don't usually do that, but they will move back and forth. And the more they will shift back and forth with that lateral movement, the more fluid the membrane is said to be. So at high temperatures, the cholesterol will help prevent that. They'll help prevent the phospholipids from moving so easily back and forth and basically making the membrane fall apart. At low temperatures, the cholesterol will help make those animal cell membranes harder to solidify, because it will make it so that the phospholipids, because you'll notice here how they're embedded between the phospholipids, it will prevent those phospholipids from packing tightly together so that the membrane becomes solid. So again, in animal cells, these cholesterol molecules, they're going to affect the fluidity or the movement of those phospholipids within the membrane. When it's a really high temperature, they actually keep them from moving so the membrane doesn't fall apart. When it's a really low temperature, they will keep the membrane from basically freezing solid, keep those phospholipids from packing so tightly together. Your glycolipids and your glycoproteins that you find within the cell membrane, they are going to be carbohydrates attached to lipids and carbohydrates attached to proteins. That's where the glyco part comes from. Right? The glyco part is going to represent a carbohydrate chain. You can see a carbohydrate chain here attached to a lipid. You can see a carbohydrate chain here attached to a protein. So a glycoprotein, carbohydrate attached to a protein, glycolipid, carbohydrate attached to a lipid. So these glycolipids and glycoproteins, their main job is cell-to-cell recognition. You can think of it almost as like it's a name tag for the cell. So these glycolipids and glycoproteins, they help mark the cell. And they help say, okay, this is a immune cell, and this is a muscle cell. Okay? But that's what they are good for. They are, in particular, very good for cell-to-cell recognition. All these molecules together are going to result in what we call our fluid mosaic model. Okay? And that fluid mosaic model is a mix of all these molecules together, okay? And it's due to the fact that you have this phospholipid layer, phospholipid bilayer, you have these proteins embedded in it, glycolipids embedded in it, steroids embedded in it, glycoproteins embedded in it. Okay? So the membrane itself is fluid. Remember, the phospholipids can move, can scoot back and forth. So the membrane itself is fluid, but it has this mosaic pattern, like a tile pattern, because it has these various molecules embedded within the phospholipids between them. So this fluid mosaic model structure of a cell membrane is what allows it to do its job. And its main job is to prevent anything and everything from just coming into the cell whenever it wants. Okay? Its main job is to provide a separate internal environment that's going to be different from the external environment. So it's going to help establish this, and it's going to help maintain it. It establishes it by actually forming the physical barrier, and then maintains it by only allowing certain things to move back and forth. And that's all going to be due to the structure. So the membrane structure, that phospholipid arrangement, is what's going to allow for that selective permeability. And so small nonpolar molecules, so tiny little molecules that don't have much of a charge, okay, that don't have any charge, those molecules can get right through. They can just slip through those phospholipids. Okay? Those phospholipids aren't interested in them because they have this large nonpolar area in the middle, so they're not interested in kicking them out. The fact that they're small allows them to slip right through. And so things that would be able to do that would be like nitrogen gas, oxygen gas, CO2. So these tiny molecules that don't have any charge, they can slip right on through. Okay? Hydrophilic substances, so substances that have a charge, they will use channel and transport proteins to get through. So you can see on this picture here, there's a channel protein here. Okay? And so these molecules will go through those channel proteins. And those are going to be things like water needs those. Larger molecules and molecules that are going to be polar, because that would make them hydrophilic, they're going to need the help of these transport or channel proteins to get across. Okay? Basically what will happen with these is this inner hydrophilic area here will kick those molecules back out, and it won't let them cross the membrane. So they need to go through these channel proteins, which will have a hydrophilic channel. So they're attracted to the sides of the channel here. And it'll have a hydrophilic channel here that will prevent that hydrophobic region of the phospholipids from kicking them back out. And now they can tunnel through the membrane to the other side. Transport or carrier proteins, like you can see here, transport or carrier proteins will change shape, and that will allow the molecule to move across the membrane. For instance, this one here, if a molecule came and joined here, it would then open up at this end to allow the molecule to pass all the way through. But again, it's doing the same thing. It's blocking out that hydrophobic region of the cell membrane so that the molecules don't get kicked right back out. Both of these kinds of proteins are specific. So it can't be any molecule that wants to go through this carrier protein that can get through there. And it can't be just any molecule that decides to go through the channel protein can get there. Okay? The proteins are going to be carrier specific. For instance, you'll have a particular channel protein called an aquaporin. Water is a larger polar molecule. So in aquaporin, those are channel proteins specifically for water. So this helps maintain that selective permeability. Any molecule can't just come back and forth through these channels. The channels are still selective and pick and choose what can come through. Smaller polar molecules, some of them can get through very, very slowly. A smaller polar molecule or a larger uncharged molecule, some of them can get through slowly, but many of them will have, for instance, like water, will have that aquaporin that allows it to move back and forth quickly. Like water can eventually sneak its way through because it's not super, super huge, but in the amounts that your cell usually needs to transmit back and forth, your cells have aquaporins that allow it to move faster. Glucose would be another example. Glucose, it doesn't have a charge, right? C6H12O6, but it's a larger molecule, so it can slowly go through, but it's not slipping back and forth quickly like those gas molecules were. So in addition to the cell membrane regulating what can go in and out, certain cells are also going to have a cell wall, right? Plants, some protists, okay? Some fungi, some prokaryotes, right? Not all prokaryotes, but some of your prokaryotes are going to have a cell wall. Remember, none of your animal cells will have this cell wall, but your all your plant cells will, some of your protists, some of your prokaryotes, and fungi will have them. And the cell wall is usually going to be made up of complex carbohydrates. Okay? If you look at what you have happening here in this cell wall structure here, you can see cellulose microfibrils here, and those cellulose microfibrils are laying down the foundation for that wall. So they'll be made of complex carbohydrates. In this particular instance, this is a plant cell wall structure, and you know it because of the particular carbohydrate, which is cellulose. The different bacteria will have, their cell walls can be made of different things. For example, peptidoglycans, a lot of times make up bacterial cell walls. Fungal cell walls are going to be made of chitin, which is a different kind of complex carbohydrate. So it depends on what cell it is as to which carbohydrate is making up the cell wall, but all cell walls will be made of, as a general, will be made of carbohydrates. So the main purpose of the cell wall is not necessarily to prevent things moving from back and forth. It is to provide structural boundaries, okay, to give it to some support. Think about the organisms that have a cell wall. Fungus, plants, they don't have skeletons. And so this cell wall is going to help provide protection. It's going to help maintain shape, and it also helps prevents the cell from taking up too much water, because as this central vacuole here fills with water, it starts pushing against the sides of the cell wall. And so that will stop the uptake of water as that pressure inside of the cell increases. So structure is more the main job of the cell wall. protection, maintenance of shape, preventing that excess uptake of water. Okay? It will provide some of a permeability barrier for particular substances, but its main job is not to be filtering things out. That is the cell membrane. And cells that have a cell wall, they still have a cell membrane right here on the other side of the cell wall too. So a cell wall does not take the place of a cell membrane. A cell wall is in addition to a cell membrane. So all kinds of cells, regardless, are going to have a cell membrane. But some will have this cell wall on top for extra structural support with it. Okay, so our focus is going to be the cell membrane and cell transport. We're going to start some labs that are going to talk about cell transport. We're going to move on to surface area and volume and do some labs with that too. But it's all going to come back to this basic concept of this phospholipid bilayer regulating what goes in and out of the cell so that the cell can maintain its internal environment. And that's why things are going to be moving back and forth across that membrane, is maintaining that internal environment.